Good morning to all. I am Aheria from Department of Microbiology and Cell Biology, Indian Institute of Science. And today in this short talk, I will be discussing some of the fundamental findings that we have come across that helps in understanding the T cell biology at the immunological synapse. So T cell activation is typically characterized by interaction of the T cell receptor with the peptide antigen on MHC on the antigen presenting cell. This leads to formation of a cell-to-cell interaction between the T cell and the antigen-presenting cell known as the immunological synapse. This synapse is characterized by actin enrichment and signalosome activation and finally downstream calcium flux. Post-synapse, when the T cell dissociates from the cognate antigen-presenting cells, there is a significant surface T cell receptor down modulation. And it has been shown by flow cytometry experiment that only 60% of the receptor is down-modulated, while the rest 40% is somehow rescued, and the down-modulated receptor is targeted for lysosomal degradation. Now, when people observe the synapse, which is a planar structure using a supported lipid bilayer, and you're observing the end phase, you observe that TCR microclusters all migrate to a central actin clear zone on the cell known as the CSMAC. This retrograde motion of TCR microclusters is seen in Jurkatz, which is a cell that has been used for most of these studies, which is a T cell line. And it is shown that this retrograde movement of TCR microcluster is mediated by a retrograde actin flow. And if you block this retrograde actin flow by means of any substrate that is on the cover slip, that also blocks the microcluster migration. But here comes the paradox. In flow cytometry, we see only 60% of the receptor is down modulated in case of primary T cells, while 40% is likely rescued. While in microscopy data using Jurkat T cells, all TCRs travel to the central CSMAC zone, which is a signal dead zone. So now the question arises, do all ligated TCR microclusters get delivered to the CSMAC? Clearly not, because there is a 40% which is not downregulated. So what happens to those 40% receptors and what might be the sorting mechanism? Keeping these questions in mind, we observed the immunological synapse in phase view using a supported lipid bilayer. And this time, we used TURF-SIM microscopy. Now, the advantage of TURF-SIM is it provides a very high spatial, temporal, and most importantly, time resolution. We used two systems of cell. First is the classical Jurkatz, where green demarcated in actin and red the TCR microclusters. And the second system that we used was the primary mouse T cell, which has been less commonly characterized at this time and resolution before. Now, just simply looking at it, though both of them are T cells, there is a significant difference in their actin architecture, which has been reported by my supervisor in her Journal of Cell Science paper 2019. Now, this time, we looked at the TCR microcluster. And we used TrackPy algorithm to detect these microclusters. So anything motile within the subsynaptic field was detected as a microcluster, and we tracked their movement along the movie. And in Jurkatz, if you see it, as expected, all the microclusters moved to the central centripetal zone. But the surprise came when we quantified these primary T cells. There was actually a significant fraction of microclusters that instead of going to the central CSMAC zone, was going to the peripheral zone in the anterograde direction. Upon quantification, we found almost 40% of the TCR microclusters travel to the periphery. And this fraction is apparently non-existent in Jurkatz or at a very low ratio. And these anterograde microcluster speed is independent of the Jurkatz speed or the speed of the inward moving microcluster, their intensities or their normalized radial position within the synaptic zone. So the question was, what might be driving these few fractions, 40% of these microclusters to the periphery? Now, as I discussed previously, the centrally moving retrograde microclusters are brought to the central by actin flow. And this is by means of frictional coupling. So we wondered, what if actin also has an anterograde fraction? We quantified the actin flow using particle image velocimetry, and we found that indeed there was a anterograde moving actin flow. It was not like a flow, it was more like these waves or bursts of actin in the outward direction, and we call them actin waves. If you look at this particular image, this is temporal color coding. So if it's a whitish tinge, it means all the colors are coming together, and it means it's a more static structure, which is seen in case of Jurkatz. While in case of primary, the structure is much more dynamic, showing that these actin waves not only contribute to the structure, but also the dynamicity at the synapse plane. Now, since we know there is an anterograde TCR cluster, there's an anterograde actin wave, we wondered if they actually co-localize. So we used an edge detection algorithm to find out the high intensity actin waves, and we superimposed that on the TCR tracks. And if the TCR tracks were within 300 nanometer radius of these actin edges, we scored them as co-localized. And indeed, we found co-localization of the actin waves with these TCR tracks. I'll show this quantification data in the following slide. So with this in mind, we now know that these TCR tracks do co-localize with actin waves. But what might be the mechanism of sorting? So to understand that, we use the aid of computational modeling. So for computational modeling in a subsynaptic zone, we define 100 tracer particles, which are dimensionless particles mimicking our microclusters. And all of these tracer particles were given a default retrograde, CSMAC directed flow. Now, in our first mode, we took those edges that we had detected from the raw data, put them on the tracer particles, and if these tracer particles were coming in contact with the waves, they were drifted to the periphery. This was our first possible mechanism. The second mechanism was there is no such thing as waves, everything is an actin flow. Whenever these retrograde moving clusters come in contact with the whirlpool of actin waves, they are just following the arrows and drifting to the periphery. To see the goodness of fit, we plotted the probability distribution and the outward time distribution of these microclusters. So the red line shows the experimental microclusters and their probability distribution with time. The blue line shows how the tracer particle behaves in mode 1. So it was beautifully matching with experimental data. Now for mode 2, all these green lines are mode 2, but with different velocity thresholding. And it was found that only if the velocity threshold is set between 13 to 33 nanometer per second, we were able to see, goodness of fit at this range. So from this, we could conclude that most likely there is these actin wave-like structure with a very limited velocity range, which might be driving these microclusters to the cell periphery. Now, one important thing about this model was that all these microclusters had a by default retrograde flow. Yet when they were coming in contact with the waves or flows, they were changing direction and going to the outward direction. That is not possible simply by frictional coupling. So that requires some high affinity molecular coupler to play a bridge between the microcluster and the actin waves. So we screened a number of molecules which has the property of binding both to actin and to the signalosome complex of TCR. And the significant one was Wiskott-Aldrich syndrome protein, or WASP. So WASP is a well-known actin nucleation factor. It binds with actin during synapse, and T cell activation also binds with the TCR signalosome. And when we knocked out WASP from the T cells, we could see that the percentage of the T cell receptor that was traveling anterogradely went significantly down compared to control. As a control, we also use CK666, which is an ARP23 inhibitor, another actin nucleation factor. There we did not see any difference. But the most startling feature was that even though that the TCR microcluster was hampered in WASP knockout, the actin waves in the anterograde direction remained unaltered. So this shows that there's a possibility that WASP could be acting as a coupler. So we use the WASP knockout cells and use the same co-localization algorithm on them, and indeed, there was slippage between the TCR tracks and the actin waves, showing that now the co-localization has gone down significantly in WASP knockout condition compared to control. So this further proved our point that WASP or any of the constitutive WASP interacting proteins could be acting as a coupler. So with this, I come to the summary of my talk. So what we found in this particular project was that in primary T cells, there exists a significant fraction of anterograde moving TCR microclusters, which are associated with these anterograde moving actin waves. And the interesting feature was that these actin waves coexisted with the vectorially opposite actin flow in the same subsynaptic plane. And somehow, the TCR microclusters are coupling with the help of WASP to these outward moving actin waves, and which is a likely mechanism of signal rescue at the T cell synapse. Interesting fact, we don't know how these waves generate, what might be the cytoskeletal mechanism behind it. And furthermore interesting, Jurkats do not have these anterograde waves. Hence, it does not have an anterograde fraction. The reason why, despite it has been in the literature for so long, most of the studies miss these outward moving clusters, which is a mechanism of receptor recycling. With this, I'd conclude my talk and acknowledge my supervisors, Dr. Sudha Kumari, my collaborators, Samuel and Dr. Sumanta Sarkar, my lab mates, interns, staff, funding, and thank you very much.
Very nice talk. Do you have an understanding of why there are actin waves? I mean, is it a polymerization, depolymerization?
No, it's not a polymerization, depolymerization. If we block polymerization or enhance polymerization using inhibitors, both the waves and the inward flow is affected.
So why waves?
So I cannot comment on it.
It's an excitatory system? Okay.
Yeah,
okay.
I mean, I really don't know, but probably there is some role of other, like some motor proteins like myosin involved in it. But the exact mechanism I cannot comment on.
But you could do some knockouts of the myosin and check.
So just to respond to Vijay's question. I think it's clear that if you allow for turnover of myosin and actin,
yeah,
then this drives the system into an excitatory system. So you get these actin waves,
yeah,
as you say.
yeah.
But so I think one can understand that. But the question is, why don't you see it in Jurkat? What is the...
So Jurkats in general are much bigger in size compared to primary T cells. So that's one thing that could be, even if that whirlpool-like motion is there, it's probably not enough to go in the outward direction. And the second is all Jurkats, since it's a cell line, it's immortalized, it has a lot of genetic alterations within itself. So we don't know what is messed up there.
Have you compared any other cells to see that, can you sort the types of cell culture people are using that lack this movement or others that have it?
I mean,
I'm sure people study Jurkat because it's convenient, but
yeah,
the reality is you should be studying primary cells, is what you're saying, right?
I do agree. But again, Jurkat is an indispensable system because of the ease with which. So we're not saying that Jurkat should not be used. But if you see something in Jurkat, just validate it once because it might be showing you the half story, not the full story. That's what they. And yes, we have tested. So this was CD8 data. We also observe CD4s, and they also have these outward motions. We have not looked at any other immune cells because they don't have this fast dynamics typically at the synapse. But yeah, it's an interesting thing to look into.
Any more questions?